Good morning to all. So today I will share the topic erythrocytes, particularly what are erythrocytes, what are their normal values, how they are formed, means erythropoiesis. We know our blood has some cellular elements, okay, and among these elements, that is leukocytes, erythrocytes, and platelets. Largest number is erythrocytes. See, what is the normal value of erythrocytes in blood? 5 to 6 million per cubic millimeter or per microliter. Whereas, what is the thrombocyte count? 1.5. Lesser than erythrocytes, and WBC is much less. Okay. Why these cells are called erythro? Erythro means red color. The sites, when cells which are red in color, we term them as erythrocytes or red blood cell, red blood corpuscles. Okay. So what are erythrocytes? Erythrocytes are red colored, disc-shaped, circular cells of the blood, and one of the most important feature is they are not having any nucleus. So you may be surprised. We know the nucleus is the it controls the cell. So without nucleus, how it can live? How it can perform its functions, isn't it? No nucleus means there will not be no formation of proteins. Because what proteins will be formed, it is determined by the nucleus, by the chromosomes, by the DNAs. Okay, in the coded form it is written there. So this code has to come to the endoplasmic reticulum. And since there is no nucleus, no mRNA will be formed. So what's the use of endoplasmic reticulum? So definitely it will not be having endoplasmic reticulum. And one important thing is they are not having any mitochondria. So we know mitochondria is the powerhouse of the cell. Here most of the energy are formed. So how it will derive its energy? We know. We have studied in biochemistry or in your class 11-12 days. There are two types of metabolism: anabolism, catabolism. For carbohydrates, the catabolic pathways are what? Glycolysis, TCA cycle, and electron transport chain is there, oxidative phosphorylation takes place there. So these are the pathway from which energy is derived from carbohydrate. From lipid, we know lipid can give us energy. So lipid also can provide energy. Yes or no? Yes or no? Whether they are able to provide energy. No. They can't. What is beta oxidation? Have you learned? Have you heard that this path? Beta oxidation? Hard. So it is the oxidation of fatty acids. And that takes place in mitochondria. So there is no mitochondria. So there will not be any beta oxidation. And fatty acid comes from fat. After hydrolysis of fat, we will get glycerol, you will get fatty acids. So these fatty acids can give us energy. Glycerol also can give us energy by the process of gluconeogenesis. You heard the name? Gluconeogenesis path? From which these are the gluconeogenesis is the path by which glucose is produced from non-carbohydrate substances. So one of the substrate is glycerol for this. So this glycerol can also be used, but our red blood cells, they cannot. Why? Because mitochondria is absent. Where the beta oxidation will take place. Let us see. We have already told the shape of the RBC is biconvex. It's like disc. At the center, it is compressed, indented from both sides. So it is almost flat. Only thing, the center is indented biconcave. Size, if you take it laterally, hello? If you take the dimension from here to here, what would be the diameter? 7.2 to 7.5, 7 to 7.5. Okay? And at the center it is thin. Flat, compressed from this side, from the bottom side, from the upper side. So here thickness will be less. It is 1 micron. How much? 1 micron. So size of the RBC is on an average it is 7.2 micron, and at the center it is 1 micron. Good. Normally there is no variation in size and shape, very little variation, 7.2, 7.5, like that only. If you take 100 ml of RBC, each RBC is having hemoglobin. So total how much hemoglobin you will get from 100 ml of blood cells, RBCs? You will get 34 gram of hemoglobin. Its outer surface is negatively charged. Is it helpful for the RBCs? Hello Ji? Is it helpful for the RBC to become negatively charged? You are sitting, no? And nobody is displacing each other. Okay. Why? There is no charge. If you had positively charged, or all of you had positively charged, then what? You will be repelling each other. As a result, what will happen? You will be in a state of movement. RBC, with the help of this negativity, they can remain in suspension. It is not sedimented while in circulation. So if I add some positively charged ion, what will happen? This will be neutralized and you will not be moving, you will be settling down. And who is this fellow? Who can do it? This fibrinogen. When there is excess fibrinogen, more than normal, it is going to do that. So there will be rollo formation. Stack of RBCs will be there. So because of this rollo formation, they will be sedimented. And they are now unable to be in suspension. This is the first time. Next time I will not allow. The membrane. What is there inside the cell? Bag of fluid with dissolved substances. It has to survive for 140 days. So before the extrusion of nucleus from the cell, they have provided you with this. You have to survive for 140 days. I am giving it now. You will be using. Every substance has got a life. All proteins has a life. How much life? We expressed it by half-life. By half-life. So all those substances will also be having half-life. So after some time they will be diminished. So if they are diminished, the enzymes are diminished. Enzymes for what? RBC is doing the functions, no? So for these functions it need energy. So energy should be there. And this energy will be coming through anaerobic pathway. We know glucose can be oxidized in our body anaerobically, also aerobically. When oxygen is there, we can transform glucose into pyruvic acid. Pyruvic acid can be converted into lactic acid in the cytoplasm, but when oxygen is there, it will be entering into the mitochondria where it will be oxidized through TCA cycle. So if, see, lot of oxygen is entering into the cell, this is the aerobic condition. Even then, pyruvic acid is converted into lactic acid. Why? Because there is no mitochondria. Where it will go? So pyruvic acid is converted into lactic acid. So in this way, RBCs develop their energy through anaerobic pathway, means anaerobic glycolysis. The membrane, the plasma membrane, what is there in the membrane? What are the constituents of membrane, plasma membrane? Protein is there, lipoprotein is there, lipid bilayer. Actually, lipid bilayer is there. And these lipids are what? PL, phospholipid. Phospholipid. Lipids, phospholipids. Okay. In between, these are the proteins. This is the fatty, actually, these are the fatty acid. And is a phospholipid phosphate part. And these are the fatty acids. And sometimes these fatty acids are, it may be saturated fatty acid, it may be unsaturated fatty acid. It may be long chain fatty acid, it may be short chain fatty acid. Long chain fatty acids. These proteins, different types of proteins are there in plasma membrane. Devolina, you studied it. You studied plasma membrane. I have seen your notebook. Can you tell some functions of these plasma proteins? Good. Sit. Through this protein, some proteins are channels, ion channels. Those ions which cannot pass easily through the membrane. So for them, some channels are there. Through these channels, sodium channels, calcium channels, potassium channels, different channels are there. And some channels are there, some transport proteins are there. They will be helping transfer of certain substances, required substances from outside the cell into the cell. Some are protecting the cell, residing at the outer surface. Some are binding sites, because some hormone will be acting on this. So these hormones has to bind with the receptors present on the surface of the cell membrane. So first it will bind with this protein, and this protein will make some changes in the inner layer. The protein is also having, is projected to the inside, and here it will cause some changes. And with these changes, it will produce second messenger. First messenger is the hormone. It is sending some message to the cell what to do. Instruction. Sending certain instruction to the cell, to the nucleus, to the cell. So what has to be done? In this situation, what has to be done? So it has attached to the, initially it came through blood and attached to the receptor, then produced some changes, produced a second messenger. Now the second messenger will produce series of events. Series of events depending on the type of hormones, type of its function. So accordingly, it will produce the changes within the cell. Though then cell will alter its function. So this is the basic mechanism. Some steroid hormones like estrogen, progesterone, adrenal cortical hormones, testosterone, they are lipid. They are made from cholesterol. Cholesterol is part of lipid. So they are, since this layer is also lipid, so when they want to enter, easily they can enter. Nobody is going to obstruct. Where is the receptor site? The receptor site for those hormones are inside the cell. But for protein, those who are protein in character, those who are, the hormones which are protein in character, biochemically, say for example, insulin, glucagon, prolactin, gonadotrophin, what is this? Somatotrophin. So these are protein in nature. So they cannot enter directly. So first they have to bind with the receptor, then they will produce the second messenger, then second messenger will do the necessary things. Clear? So, and Debolina, the cell membranes has got some flexibility, isn't it? It is not rigid. Some cells are like RBCs. The cell membranes are flexible, not rigid. And this rigidity or flexibility depends on this Bodulum. Who is it? Cholesterol. This is the cholesterol. The amount of cholesterol, if it is more in the lipid bilayer, then its flexibility will be more. 35%, 45%. And it is the person who is creating this flexibility. But in other cells, for example, epithelial cells, surface cells. So they are this type of the cells here. They are under various pressure, various external pressure. So they should be tightly packed. They should be tightly packed. Their outer membrane should not be flexible. So here you will find less. Here you will find not 45%, very less amount of cholesterol. So less cholesterol, rigidity. More cholesterol, flexibility. I think we will not forget this. See, another thing is helping the RBCs. Hello, one question I left to you. The question was that capillary diameter of capillary is lesser than the size of the RBC. Everybody listen. There may be short question. MCQ. Diameter of RBC is more than diameter of the capillary. Then how this RBC travel through the capillaries? Yeah, they can shape their chain. They can shape their chains. When they are coming there, so they change their, like amoeba. They should change their shape. Okay. And who is helping in shaping the chain, changing the shape? These are the things. What are these? These are cytoskeletons. What are these? Have you heard the name? Hey, you have the skeletal structure, no? That's why you are having a shape. Otherwise, you would have been jellyfish. No definite shape. So you have got a definite shape because of what? The skeleton. It is giving you a particular shape. Your bones at your face, at your head, giving you a particular shape. Okay. So this pectine, actin, we find actin also in muscles, actin, myosin. When they slide together, the muscle is shortened. Okay. So, and chitin, these are the skeleton of cell, any cell. All the cells are having cytoskeleton. They maintain the shape of the cell. When it is required to change the shape of the cell, then this pectin, actin, and chirin, these cytokines contract. As a result, they can change the shape. Is that clear now? Everybody understood? Okay. What is the water content in RBC? 62.5%. Hemoglobin occupies 35%. And there are other substances. As I said, they have to survive for 140 days. So for their survival, some necessary things are there. And what are these? Glucose, lipids, cholesterol, lecithin, proteins, glutathione. I marked glutathione. RBC is under lot of oxidative stress. And because of this stress, the cell membrane may be ruptured, the cell may die because of this stress. Because moreover, it is carrying lot of oxygen. The stress is more. And moreover, we are, for so many reasons, we are taking antibiotics. And these antibiotics are also producing stress. This type of stress. As a result, it has to face the lot of stress. To do my function, to carry oxygen and carbon dioxide, I have to remain fit. So who is protecting me against this stress? Sorry. It is glutathione. It is protecting. When we lose this protecting activity, the cell will die. When the amount of carbonic anhydrase, these are protein. These enzymes are protein. This is protein. The glycolytic enzymes. I will ask. Let's see. Any one enzyme of the glycolytic path? Everybody learned glycolytic path. Isn't it? Everybody learned. And it was not learned by heart. Huh? Huh? Hexokinase, glucokinase, fructose 1,6-diphosphatase, lactose dehydrogenase, pyruvate kinase. A lot of enzymes are there. So these enzymes are there. It is given. So you now survive. I have given you certain amount of enzyme. So with this you derive your energy. Okay. But these enzymes, since protein in nature, they have got some half-life. So after some time their life will be gone. So the cell will die. That's why it is 140 days. There are also some. We have blood groups. We group our blood into A, AB, O, or B. These are the blood groups depending on the antigen present on the surface membrane of the cell RBC. If I have antigen A, my blood group is A. If I have B, my blood group is B. If I have both, I will be having AB. If I don't have anyone, then my blood group is O. So these antigens are present on the surface. As I said, their energy requirement is less and they derive their energy from anaerobic glycolysis, and there is another path, HMP shunt. Heard the name? SMP shunt. Then you might have heard this pentose phosphate pathway. Okay, glucose is hexose. Hey, what are carbohydrates? What are carbohydrates? Tell me, who can tell? Carbohydrates are polyhydroxy alcohol or ketones. Lot of hydroxyl groups will be there, lot of carbons are there, lot of hydrogen is there, lot of oxygen is there, but usually oxygen and hydrogen will remains in the ratio of water, H2O. And whatever the amount of water, that will be the amount of carbon. Cn, n number of carbon. So H2O will also be n numbers. So these are actually polyhydroxy aldehyde or ketones. Everything will be fine. Aldehyde or ketones. Say monosaccharides, glucose, this is aldehyde. Say fructose, there is no aldehyde group, there is keto group. So this is fructose sugar. Okay. Sucrose, chini, sugar. So there is a disaccharide. One is glucose, another is fructose. But both the groups. The glucose, it is having aldehyde group, fructose is having keto group. This keto group, both the aldehyde and both are reducing group. They can reduce. It has got the reducing power. But this group remain attached to each other. So it lost their reducing property. Now this disaccharide is a non-reducing sugar because they are not having any reducing group, free reducing group. Whereas lactose, fructose, maltose, these are reducing sugar. They can reduce Benedict reagent, but sucrose can't because there is no free aldehyde or ketone group. Okay, that is not the subject. So glucose is a hexose. The monosaccharides are classified depending on number of carbonaceous atoms present in it. If it contains 3 carbon, we call it triose. If there is 4 carbon, we call it tetrose. If there are 5 carbons, pentose. Like that's hexose, heptose, octose, nanose. We need pentose. We need pentose. We need pentose sugar. For what? What is DNA? What is the full form of DNA? Deoxyribose. This is a pentose sugar. And RNA ribonucleic, that is ribose. So all these nucleic acids require pentose. Who will supply this pentose? Either you have to eat pentose or it has to be produced in my body from glucose. That is the pathway. Not only this, it has some other functions. So you have seen this many times. Our purpose is erythropoiesis. To learn, we will learn erythropoiesis. Whatever I taught since beginning, is every point is important. So try to remember those points. And while we are, for everybody, listen carefully. While we will be going through the process of erythropoiesis, you have to observe what is happening. Minutely, what is happening? What is happening to their size, from which it is developed and ultimately what is produced. In between there will be so many cells. So what happens to the size? What is coming in, entering in? What new things are added to the cells? What is coming out of the cell? Well, are there any change in its shape? So these are the points you have to observe. Then you will be understanding what happens during erythropoiesis. Multiplication but through mitosis. Initially you will observe they are having all mitotic activity, but later we will find it has lost this mitotic activity. So these are the observable, noticeable changes that take place during erythropoiesis. Erythropoiesis is a kind of hemopoiesis. Hem means blood, poiesis means synthesis. So blood synthesis is hemopoiesis. So blood, there are so many constituents. I mean, RBCs are there. So for this hemopoiesis is termed as erythropoiesis. Similarly, there is leukopoiesis. There is thrombopoiesis. So when we want to study hemopoiesis of thrombocyte, we will term it as thrombopoiesis. When we are interested to learn formation of granulocytes, granulopoiesis. Okay. So what is erythropoiesis? How it is defined? Erythropoiesis is the process of development, differentiation, because gradually they will be differentiated, new, they will be changing their identity from the previous one. So they will be differentiated as well as ultimately they will be matured. So this process of making erythrocyte from the primitive, from the ancestor, the ancestors are there, mother cell is there, from the mother cells, how they are derived, it is erythropoiesis. Regarding erythropoiesis, there are two hypotheses previously. At that time, we didn't have so much knowledge regarding erythropoiesis. So two gentlemen, they gave two hypotheses. One hypothesis tells it is monophyletic theory, and what this theory states? It states all cells derive from a particular stem cell. They are the originator. From these stem cells, all varieties of blood cells are coming, producing. So this was the theory, monophyletic theory. And polyphyletic theory, it says no, stem cells are different and this cell will produce erythrocyte, they will produce lymphocyte, they will produce granulocytes and so on. So different lines, for different lines of cells, blood cells, there are different stem cells. So this is the polyphyletic theory. Let us now understand where from the RBCs are generated. What is their site of synthesis? When we had been in the embryonic stage, so initially at 8 weeks, 9 weeks, at 8 weeks, heartbeat starts. At 8 weeks, 7 weeks, 8 weeks, heartbeat starts. That means at that time, circulatory fluid, blood has started coming. So those blood is coming from yolk sac. So this embryo is developing from 3 layers. What are these 3 layers? What are these 3 terminal layers? Ectoderm, endoderm, mesoderm. And this mesoderm is responsible for not only responsible for heart, for making heart, vascular system, blood. So it is the cells of the mesoderm. They will first start synthesizing RBCs. And when after 3 months of embryonic life, after first trimester, what happens? Liver and spleen comes into action to produce. At the time, 3 months, bone is not formed, no? When bone is started forming, at the same time it will start producing. Bone will be there. When bone will be appearing, first femur will be easily observable through ultrasonography. So femur is a long bone. So it is filled up with red bone marrow. So this red bone marrow, they are having those stem cells. So they will start hemopoietic stem cells. These stem cells, no, they are unique. In our body, so many varieties of cells, so many types of cells are there. These somatic cells, these stem cells can produce all the variety of cells. And the stem cells which are directed to or committed to formation of RBC, WBC, all variety of blood cells, they are called hemopoietic stem cells. Their purpose is only to produce blood cells. So we call them, this type of stem cells are termed as hemopoietic stem cells. Yellow marrow, red bone marrow, red marrow. After 20, see, we stop growing. After certain age, we don't grow further. We attain our maximum length. Isn't it? At what age you attain this? 25. After 18, you don't increase in size. And they don't increase their size after 16. So before the closure of epiphysis, you are continuing for some time, even for more time. And after that, you cease your elongation of your length. Why? Because of, will you increase? Increase of femur length, increase of humerus, long bones will increase your height. But these long bones cannot increase in length, they can increase in lateral size. So why it happens? Previously, from here to here, there was red bone marrow. See, when you are below 1 year, it was occupied by red bone marrow. So when you are 1 to 10 years old, this way yellow marrow is replacing this, gradually increasing. When you are 10 to 20 years, see what is happening. Very small portion of red bone marrow is left at the end. And after that, when you are more than 25, see, no red bone marrow, only their contribution is very less. So, during this period, adult period, adolescent period, childhood period, infant period, other than red bone marrow of long bones from some other place, erythrocytes are generated, erythropoiesis is taking place, the bones of skull, bones of hip bones, pelvic bones, pectoral girdles, then sternum, ribs, they are having red bone marrow. So they will continue. This is not replaced by yellow marrow, so they will continue erythropoiesis. See, they say adult skeleton, so no red bone marrow. These are the sites where erythropoiesis takes place. Coming week there is exam. Remember this. Sis, I will be asking MCQs. So all will be relevant to this. Already what I have explained, what I am explaining. So I told every point you remember. Try to remember. Immediately note it down. One simple question can be asked. Why in adult stage femur cannot produce cannot go for erythropoiesis? Simple question. Now you are able to answer this. Those who are listening, or those not listening, they will not be able to do it. Do they make the answer? What is this? The steps involved in erythropoiesis. These are to be, see, if question comes, there will maximum 6 marks. There are maximum marks for 2 questions, 10, 10. Question number 1 will be having 10 marks. Question number 2 will also be 10 marks. And the rest part will be fragmented. But 2 marks, 3 marks, 1 marks, like the MCQs are 1 MCQ, 1 mark. So in that way, you will be having 100 marks. And pattern I will be giving you later. And 1 mark, 1 question will be from ETCOM. When question will be from there, today there is, I hope, one ETCOM class is there. ETCOM means your attitude, your ethics and communication. Ethics, what would be your ethics? What would be your attitude? It will be decided by your professionalism. Because I said, you came with a particular attitude. Now, because of this profession, you will be changing your attitude. You will be changing your ethics. So, lot of development is going to happen in your professionalism. Now, there is lot of students. Again, I will repeat those class that what was there? Ethics, attitude and professionalism. What are the guidelines? It is taught in ETCOM. What do you think about a patient? What do you think about a doctor? What are your duties? What are your responsibilities? Who can be said a doctor? So your moral responsibility, what should be your attitude to the patients, how you should deal with the patients. And all the patient has got one right. This right has been given by the human right. What is the right? Whatever you are going to do with me, you have to explain. This is my right. If I ask you a question, you cannot tell, are you a doctor? You should not do that. It should not be, your attitude should not be like that. Nowadays it is taught like that. The CBME curriculum is for that. For that you should have certain particular feelings, empathy for them. That's why we are sending you to the villages, to the communities. See their sufferings. You are coming from well-to-do families, all of you. So your social, family condition, economic condition is not equal to that. They require your service. And you are to provide. That's why you are here. You are committed to provide your service to them. And how you will provide, what should be your attitude? That is determining this professionalism. I'll take another class because at that time, this lot of I think that time 20 or 30 students were there. PASC. What does it mean? What does it mean? Pluripotent. Pluripotent means multiple. Pluri means multiple. Multiple type of cells I can produce. Okay, I am the principal. Under me there are a lot of teachers, and you will be teaching physiology, you will be teaching community medicine, you will be teaching this. So I will be deciding. So it is the stem cell. This is actually the stem cell. Committed for, it will produce only blood cells. Okay. So for the stem cell, they committed for or destined for this purpose. And one thing, they are transforming, they are multiplying, they are transforming into different type of blood cells. So time will come, they will be exhausted? No. They have the power of replication or regeneration of themselves. When otherwise the stem cell will be depleted from the bone marrow. Then it is not possible. Means they have the capacity of self-generation. And at the same time, some other committed cells that cannot be transformed back into stem cells. Once you are committed, you cannot come back. Many times you can multiply, but you cannot produce the stem cells since you are committed to duality. Okay. So, PSSA, these are the actual stem cells. From there, what is burst forming unit erythrocyte? BFUE. E stands for erythrocyte. And these stage, there are a lot of factors they require because this is transforming the stem cells into committed one. So, lot of directions, lot of influences are to be made to these cells. What you will be doing? Either you will be producing erythrocytes, that guidance is given, or you will be producing granulocyte, or you will be producing thrombocyte. That information is given here only. What are the influencing factors? IL-1, IL-6, IL-3. IL means interleukin. Interleukin-1, interleukin-6, interleukin-3. Actually, what they do? When PSSC is converted into BFUE, there is lot of mitotic activity. So for this mitotic activity, when new cells are coming, so formation of these new cells will require synthesis. New cells means new nucleus, means new DNA. So stimulation for DNA synthesis should be there. So they are providing this DNA synthesis, stimulating DNA synthesis so that it can produce a lot of cells. Otherwise, it is not possible. And another factor is GM-CSF. Another factor is GM, granulocyte macrophage. Granulocyte, right? Granulocyte macrophage committed colony forming unit. C for colony, F for forming, U for unit, colonies of RBCs, colonies of WBCs, colonies of D. That's why they are called colony forming units. And erythropoietin is also essential for divisions and differentiation, transformation from this to this. What are these erythropoietin? Alpha globulin. Okay? Since these are formed from tomorrow in my this is very important. Question should be there either in university exam or in exam. Very common question. Repeatedly it comes. Erythropoietin. How erythropoietin? What is the important erythropoietin? You can consider it as a hormone. And these are formed in our body when we feel hypoxic. For erythropoiesis, the great stimulation, most important stimulation comes from hypoxia. Hypoxia means lack of oxygen to the tissues. When there is lack of supply of oxygen to the tissues, immediately the responsible cells, they become excited and produce erythropoietin. What are these cells? Where they lie? Glomerulus, proximal convoluted tubule, loop of Henle, distal convoluted tubule. Now see, lot of capillaries are this is up to this is proximal, this is the loop of Henle. So lot of capillaries are around there, around the tubules. These capillaries are called peritubular capillaries. These are peritubular. If I ask where you will find peritubular capillaries, when I will be asking what are the sites from where erythropoietin is generated? So this is one site. Which one? This is the capillary and this is the tube. So in between capillary and renal tubule, in between there is a space. Actually there is no space, vacant space. These are occupied by interstitial cells. And some interstitial cells of this kidney, they generate erythropoietin. This is one source. So interstitial cells of peritubular capillaries. Now you understand what are these? So they can generate. They are stimulated when they feel hypoxic. When the cells become hypoxic, so they start producing erythropoietin. Another side is there. See, this is the actual side. This is the tubule. These are the capillary, tubule. In between, this is the cell, peritubular fibroblast. Peritubular fibroblast. So they are actually responsible for erythropoietin. Your kidney is damaging. Sorry, not yours. One person's kidney is damaging. Okay, it happens in diabetic patients. Okay, diabetes affects kidney, heart, eye. These are the target. So first it targets to destroy the kidney gradually. So what it will do? It will gradually diminish the glomerulus. glomerular filtration membranes, so that it cannot, there will be difficulty in filtering the waste products, difficulty in the formation of urine. Okay, so under this condition, we termed it as chronic renal disease, CKD, chronic kidney disease. So in chronic kidney disease, there is anemia. When kidneys are going, then I will be facing anemia. Why? Because of loss of them. Why loss? For fibrosis, there is some fibrotic changes. These cells are occupied. The kidney cells are normal. I mean, the glomerular cells, everything are changing into fibrosed. How? These cells are transformed into these cells. As a result, the whole kidney, the functional units are gradually diminishing. So it is now unable to produce erythropoietin. So lack of stimulation to bone marrow cells, so there will be anemia. We will explain it later. There are so many factors. You are not eating vegetables. Only fish, meat you prefer. You don't eat, where is that girl? Hey, tell me how much vegetables we should eat every day? I told you. 100 gram of vegetables, 100 gram of green vegetables we should eat every day. And they will provide all the required B group vitamins. And these when you will be studying different metabolic reactions, you will find lot of vitamins in the form of coenzyme, cofactor, they are participating in the reactions. So if these vitamins like TPP, PP, in hemoglobin biosynthesis we have seen, for adding succinyl-CoA with glycine, we need pyridoxal phosphate. PP means pyridoxal, it is vitamin B6. So if there is any deficiency of these vitamins in our body, then this is hemoglobin biosynthesis has to suffer. So if you are ignoring taking vegetables, this will provide your meat, fish, they are not going to provide B group vitamins. The stem cells means these are the stem cells. So by the influence of this factor, erythro means erythropoietin, this factor and these factors, they are transformed into BFU, burst forming unit erythrocytes. Okay? Again, these will be transformed into colony forming. This is the colony forming CFU for erythrocyte. It may be CFG for granulocyte. So CFM. So in this way it may be different, but we are interested in this CFUE because from this it has been committed. It is the progenitor cells. It is the progenitor cells from which these erythrocytes will be produced.